So polypeptide synthesis is the biological production of peptides, which are organic compounds in which multiple amino acids are linked via peptide bonds. It is essential for creating proteins that carry out different functions in our bodies. For example, proteins actin and mucin build up our muscles. It is essential for creating enzymes that control different biochemical pathways happening inside the cells. For example, cellular respiration takes multiple steps which are controlled by different enzymes. It is essential for proteins control different characteristics of every living organism. Therefore, what we are overall is because of different types of protein expressions. It is essential for forming products that are necessary to carry out replication, transcription, and translation as well. Transcription is a process by which genetic information is transferred from a double helical DNA to a single-stranded mRNA. It is the first step of gene expression. It is important because transcription produces mRNA, which is necessary for carrying out translation. Transcription occurs in a structure within the nucleus called the nucleolus. The whole transcription process occurs in 3 stages, and they are initiation, elongation, and termination. In initiation, the DNA double helix unwinds, forming a transcription bubble. There are 2 separated strands, a template strand that goes 3 to 5, and a non-template strand that goes 5 to 3. The sequence in the DNA which indicates the initiation site is known as the promoter. For my sequence, the DNA start codon, or the promoter, is TAC. RNA polymerase enzyme binds to the promoter and begins to add complementary bases to the template strand. The only difference here is that in the case of mRNA, the base thymine is replaced with uracil. The next step is elongation. In elongation, the RNA polymerase enzyme moves along in the 3 to 5 direction on the template strand and adds new nucleotide base pairs complementary to that of the template strand. With the addition of new nucleotide base pairs, the RNA strand keeps elongating. The next step is termination. In termination, RNA polymerase encounters a definite DNA sequence known as the terminator, which acts as a stop signal for the termination. Once it encounters this sequence, transcription stops. In this case, for my sequence, the sequence is ATT. Translation is the next step in the process. Translation is the process by which mRNA is transcribed into protein. This involves 2 types of RNA, tRNA and mRNA. Translation takes place in the ribosome. Before translation begins, nucleotide base pairs are clustered into groups of 3, known as codons. Each codon contains 3 nucleotide base pairs formed by the combination of A, U, G, and C. To form proteins, the amino acids are brought by the tRNA molecules. In translation, the codons of an mRNA are read in order from the 5 end to the 3 end by molecules called RNAs or tRNAs. Each tRNA has an anticodon, a set of 3 nucleotides that bind to a matching mRNA codon through base pairing. The other end of the tRNA carries the amino acids that specified by the codon. The same as transcription, translation is divided into the same 3 steps: initiation, elongation, and termination. Inside eukaryotic cells, initiation goes like this. First, the transfer RNA carrying methionine attaches to the small ribosomal subunit. Together, they bind to the 5 end of the messenger RNA by recognizing the 5 GTP cap added during processing in the nucleus. Then, they walk along the messenger RNA in the 3 direction, stopping when they reach the start codon, which is, in this case, AUG. So after the initiator tRNA binds to the start codon and a large ribosomal subunit joins, this forms the initiation complex. Elongation occurs after the initiation complex has formed, but before any amino acids have been linked to make a chain. In elongation, methionine carrying transfer RNA starts out in the middle slot of the ribosome, called the P site. Next to it, a fresh codon is exposed in another slot, called the A site. The A site will be the landing site for the next transfer RNA, one whose anticodon is perfect or complementary to match the exposed codon on the messenger RNA. Once the matching transfer RNA has landed in the A site, it's time for the action. That is, the formation of the peptide bond that connects one amino acid to another. This step transfers the methionine from the first transfer RNA onto the amino acid of the second transfer RNA in the A site, which in this case is valine. Once the peptide bond is formed, the messenger RNA is pulled onward through the ribosome by exactly one codon. This shift allows for the first empty transfer RNA to drift out via the E, exit site. This also exposes a new codon in the A site, so the whole cycle can repeat. Termination is the last step in translation. The polypeptide chain keeps elongating until UAA is detected, which in this case is the stop codon. Once the ribosome encounters this stop codon, it releases the polypeptide chain that will consist of methionine, valine, arginine, lysine, and glycine. The ribosomal subunits disconnect along with the tRNA and mRNA as soon as the polypeptide chain is released. Now, once the polypeptide chain is released, it undergoes different structural modifications to form a functional protein. Now, models like this one make systems easier to interpret and evaluate, but models are often limited because they are unable to incorporate all the aspects influencing a system. In other words, some systems are too complex to be modeled in their entirety, and some of these aspects of a polypeptide chain I would not be able to understand without the explanations. In saying that, this model has aspects that are much easier to understand than words, so models can have some benefits and some limitations. So that is the presentation done. I hope you enjoy.